So, hi everyone. Today we are going to discuss NEST 2024 Biology Session 1 paper. So, let's take the first question. Okay. Okay, so we'll start with second question. Uh, so, the question says that the effect of elevated CO2 supply, presence of legume and phosphate availability on the above ground biomass production of a grassland is depicted over here. And we have to choose the correct option based on this data. So the first option says that nitrogen deprived plants grow faster under ambient CO2 condition. So if I check with the ambient CO2 condition, in this case and ambient CO2 condition in these cases, there are no direct link which associates with the nitrogen deprived plant growth, or it is directly showing the link with the nitrogen deprived plant growth, so this option get eliminated. Next option, I have the growth of the plant is enhanced under elevated CO2 condition in presence of the legume. So here I have the case of presence of legume, and when I check for the growth factor when CO2 is elevated, so elevated levels of CO2, I can clearly see that there is an increase in the plant growth. So this option goes correct. Next I have the third option, which says that in absence of phosphate, legume provides no growth benefit to the plant. So here we have the option of a legume without phosphate. So here I can see that in absence of phosphate, the plant growth is less, or the above biomass production is less. So here I can clearly see that there is an increased in biomass level with addition of phosphate. So this option also goes wrong. In the fourth option, it says that in the presence of legume, there is no growth benefit on addition of phosphate under elevated CO2 condition. This is also incorrect. I can see from the option, as the biomass level is less in both the cases. And here the biomass level is increased in both the cases. So this option also gets eliminated. So option B goes correct over here. Let's move on to the next question. In this question, it is said that there is a cross of plant, and the flower and the gene over here is the flower color, which is governed by a single gene locus. The flower can be either white, red, blue, or purple. So, however, a single flower will never have different color petals. That means they will never show a co-dominance factor, but incomplete dominance may be shown. Here I have crosses. You can go through this. Let us directly go to the options. Here I have the first option, which says that there are two alleles governing the flower color. I can clearly see that there are multiple color, more than two colors. It's white, red, blue, and purple. So for a single gene locus, there are multiple alleles. So there are not only two alleles. So this option gets wrong, eliminated. The second option says that the purple allele is dominant over the red allele, and red allele is dominant over the blue allele. So this we can check over here in the cross of red with purple, where it says that mostly red and purple flowers and some are blue flowers. So we cannot conclude from here that the purple color is dominant or recessive over red color. This can we can directly say that we cannot directly say that it's dominant. They may be incompletely dominant over each other. So this option gets eliminated. The third option says that red allele and blue allele show incomplete dominance. I'll go to the cross with red allele and blue allele. So it's over here. So when I cross red with blue color, all flowers are purple and some are red. So I can clearly see that there is a mixture of red plus blue which produces purple color flowers. So this is an indication that there is an incomplete dominance over this. So this option gets correct. And the fourth one says that white allele is dominant over purple allele, which you can check from the cross of white and purple, which is wrong. So let's move on to the next question. here we have given a pedigree analysis to analyze. In this pedigree analysis, you can see there is so the parents is generally first unaffected, and then the children are affected. Generally, this type of pattern is seen in autosomal recessive disorders. So let us directly go to the question and verify which options are correct. So yes, we have to. Okay, we have to choose the correct combination. So let's see. The first option says that if the inheritance pattern is autosomal recessive, then individual 38, so I have individual 38 is a carrier of a disease. Yes, this option is correct. So if only this individual is autosomal recessive carrier, let's suppose it's A, then there is a probability that this seventh one will be also a autosomal recessive carrier. Then only this progeny will be affected. There is a probability that this progeny will be affected. So this option gets correct. The second option says that if the inheritance pattern is X-linked recessive, so then the individual 2 and 6, 2 6 and 3 7. I have 2 6, 2 6 and 3 7 are the carrier of the disease. So let us check with this. So the individual 2 6. So this is a affect. So this is a X-linked recessive. That means this is affected with this male is affected. So as the progenies are unaffected, but that means there is a chance that the females over here are unaffected. So I can also clearly see that the female is unaffected over here. So the female may be unaffected and also be a carrier of this. So the genotype of this seventh number would be X carrier and X. So this is a possibility. Then I have an individual which is, I think, 3 7. The individual 3 7. So here I have 3 7, which can also be a which can also be a carrier. So I can clearly say that yes, this is a carrier one, and this also becomes a carrier one. Okay, there is also a probability that this 2 6 may also be a normal one. But according to option, there is also a probability that there is also a X-linked recessive associated with this particular. So this option also gets correct. The third one says that individual of 1 1, so individual 1 1 and 1 2 homozygous are homozygous for the allele under study. No, they are not homozygous for allele to study. As I say, if I if the alleles that are present over here are heterozygous, that means these should be effective. So if I have an autosomal dominant trait, then this would be A and A. So then these should be affected individuals, but they are not affected. There is a generation skip over here. So I can clearly see that the parents are unaffected, but the children are affected. So this means that there is a recessive character associated with this, which is transferred to the further generation. So this option gets wrong. Then I have the inheritance pattern in autosomal recessive. Then individuals 3 2 and 4 1 must be the carrier of the disease. So I have 3 2. So this individual 3 2 and 4 1. So they should be the must be the carrier of this disease. So if I check their parents' genotype, so I can clearly see that for an autosomal recessive disorder, this could be a heterozygous one. So to be an heterozygous, if I write about the genotype of this particular 3 2, so as it is unaffected, so it must have been this. But at the same time, if I check about the 4 1 part, there is an association of disease with the progeny from the 7 and 8, which is also a progeny from the 7 8. One is also a progeny from 7 and 8. So if I check about the genotype of this particular one, so here I can see there is a heterozygous condition. So this one, not necessarily be the carrier of the disease. It can also be a normal progeny. So this word must is not correct over here. It should be just maybe the carrier of disease. So this option gets wrong. So only option 1 and 2 are correct. So this goes with option B. Okay, so this is a question from the recent Nobel Prize awards. So that was given to Kathleen Corico and Drew Weissman. So they worked on the mRNA vaccine technique, which is generally used to treat viral diseases. So in their research work, they did work on SARS-CoV-2, which is coronavirus, by developing a novel mRNA. So how does a mRNA vaccine works? So mRNA vaccine, they introduce an mRNA into human body which code for a particular spike protein, and this is recognized by our antibodies. This recognition is now stored by the memory cells, and when we have subsequent attack of this virus again, the memory cells is activated, and killer cell that activates the killer cells and kills the organism, the pathogen or the organism. So so let's check about the questions. So it says that the pseudoiridine incorporated mRNA, but the normal, not the normal with AUGC, is better than it's better for RNA vaccine. And the reason that the pseudoiridine incorporated mRNA worked as a better vaccine, I have to tell the reason out of this. So the let's go from the last option. So the last option says that spike protein from unmodified mRNA cannot induce immunoresponse. So this option goes wrong because it is telling that the mRNA with this modification is a better vaccine. So this option cannot work over here. The third option says that antigen presenting cells can translate only pseudoiridine incorporated mRNA. No, it's not like that. Antigen presenting cells can translate mRNA with normal basis also. So this option also gets wrong. The second option tells that the unmodified mRNA encoding spike protein cannot be translated by the ribosome. So this is also wrong because unmodified mRNA is also present in our body, and that will also encode a spike protein which can also be translated by our ribosome. But the thing is, when pseudoiridine is incorporated into mRNA, the vaccine works better. So there is a better recognition of the spike protein. So the first option says that the innate immunity system does not recognize the pseudoiridine incorporated mRNA, allowing translation of the spike protein. This option is relatively correct in compared to the other three options. So this option gets correct. So option A is correct for this question. Let's move to the next question. Oh, this is from Messelcell-Stahel experiment. So this question says that I have an organism that utilizes NH4Cl as a nitrogen source, was grown for a several generation for 4 different conditions as given below. So there is an organism which can utilize NH4Cl as a nitrogen source. So basically, bacteria use nitrogen source for preparing their nucleotides that will be helpful in DNA replication. That will be help in making of new strands. So I have a different growth condition over here. In the first medium containing NH4, so there is a 15th level heavy nitrogen, and the organism is grown over this medium. Then I have a medium containing 14th NH4Cl. Then there is another condition in which first I am culturing it in 15 NH, and then I am transferring it to 14 NH. And again I have another condition in which I am putting it in 15 NH, and then I am transferring it into 14 NH, and I am having a multiple generation for this. So let us analyze this question. So I have a DNA isolated from this by density centrifugation. So in Messelcell-Stahel experiment, we basically use cesium chloride density centrifugation. But there is a change in this question, is that the question is saying that we have to assume that the mode of DNA replication should be a dispersive mode of replication. It should not be the semi-conservative mode of replication which is generally taking place, which was hypothesized by Messelcell and Stahl. So this is a modified, or this is an a hypothetical modified Messelcell-Stahl experiment. So basically, what dispersive medium says that if there are two strands of chromosome, the strands of the chromosome in semi-conservative mode, what happens is the both strands separates out and a new strand is generated over the strand. But in dispersive mode, what happens is if there is a two-strand DNA, double-stranded DNA, then a fragment of this DNA is generated, and the other fragment is generated from the complementary base pairs from this sequence. So there is a dispersion over here. So there is no complete strand is synthesized over here, but there is a discrete strand synthesized over here. So you can see that the complete strand is not from the parent, rather the strand contain half from the parent and half from the newly synthesized. So you cannot say that half, that doesn't mean that the new whole strand, complementary strand, but the half of a particular single strand. So you can say that half of the has coming from the parent and the other half is coming from the new strand, newly synthesized DNA from the DNA polymerase. So if I follow this method, then I would assume, what would I get over here? So if I go to the option, this option, so in when I grow in a N15 medium, so this says that I have grown only in N15 medium, so I'll get a hybrid band. So there will be a mix of N14 and N15. So here, but the initially I have no indication that what the organism has been grown on previously. So directly I will consider, let's suppose NH4Cl on the 15th isotope. So I can get directly I will get a heavy band because I have grown it for a multiple generation. And here the generation is also not mentioned over here, how many generation I have replicated. So I'll get a heavy band over here. In the second question, in the second option, it says that I have grown in a N14NH4Cl. So there will be a light band over here because everything will be. So let me change the color of the pen. Okay. So I have a light band and a let's suppose I have a heavy band and I have a heavy band and a light band. So it's a dispersive medium, so I will have in such a fashion this, then this. So it will be something like this. So there will be half of blue color and half of red color. So let me, so like this, somewhat like this will be somewhat like this. So it will be a dispersive mode. So I will get so for the condition of medium containing whole NH4 14 NH4, so I will get a lighter band over here. So I got a lighter band over here, I got a heavy band over here. Then what it is saying that I cultured N15NH4Cl by culturing in medium containing, so medium containing followed by culturing in N14NH4Cl. So first I cultured in N15, then I cultured in N14. So I will get a medium range. I will get a hybrid of these two, what I have shown over here. So I will get a hybrid of the both of the DNA strands. So it will be some strand will have a N14 level, some strand will have a N15 level. So this shows the hybrid structure over here, hybrid DNA over here. So this is the density centrifugation one. So heavier will be at the down position and lighter DNA will be at the above position. So this is the lighter one, this is the heavier one, and this is the hybrid one. So this option goes correct for this question. Moving on to the next question. Okay, this question is from photorespiration chapter. Photorespiration chapter. So in photorespiration, one molecule of 3-phosphoglycerate from ribulose bisphosphate So this is so in photorespiration, 3-phosphoglycerate is produced from one from ribulose bisphosphate. So during photorespiration, what happens is one molecule of 3-phosphoglycerate and one molecule of 2-phosphoglycolate are produced from this ribulose biphosphate. So there is so I have ribulose bisphosphate, and this will yield me 3-phosphoglycerate. So I'll write it 3-phosphoglycerate plus 2-phosphoglycolate. So this is the whole reaction over here. So RUBP produces 3-phosphoglycerate and 2-phosphoglycolate. But the thing is, this photorespiration is very much wasteful process for a plant because it neither produces ATP nor NADPH. So this is what basically the photorespiration is. And if I talk about the location where photorespiration occurs, is the phosphoglycolate is initially converted to glycolate in chloroplast, but not in peroxisome. Then the glycolate is transported to peroxisome, where the further metabolism occurs. So if I come to the options, the first option says that photorespiration produces one molecule of 3-phosphoglycerate from ribulose bisphosphate. This option is correct. Then I have photorespiration is a wasteful process. Okay, this option is also correct. The third option says that phosphoglycolate is converted into glycolate in peroxisome. No, this is wrong. The phosphoglycolate is initially converted to glycolate in chloroplast, but not in peroxisome. So this option is wrong. The fourth option says that photorespiration in C3 plants is less compared to C4 plants. This is wrong. The photorespiration occurs in C4 plants. So the option A is correct because option 1 and 2 is correct. Moving on to the next question. Okay, so this question is from carbon fixation. So it says that in order to generate a crop variety with C4 carbon fixation, a researcher expressed pepcase in enzyme, pepcase enzyme from C4 pathway in leaves of C3 plant. However, the modified C3 plant did not show any carbon fixation efficiency as expected from a C4 pathway. The most likely reason is this direct question from NCRT, which says that there is a need of compartmentalization for pepcase to be active. So that's what happens in the C4 plants have two compartments where the cycle gets the C4 cycle gets happen. So here I have the first option, which says that an improper compartmentalization of C4 peptidase in C3 leaves. So this option is correct. The other options is wrong because the peptidase from C4 plant is catalytically inactive. So this is wrong. Obviously, this should be active. Carbon dioxide fixed by C4 pathway cannot be utilized by Rubisco of the C3 plants. Not such thing happens. Overexpression of peptidase from C4 plant increase photorespiration. No, this is also wrong. So the option A goes correct over here. Moving on to the next question. So there's a question from the molecular weight from the sediment coefficient of ribosomes present in eukaryotes and prokaryotes. Let me clearly say that this is a purely memory based question. If someone has remembered this particular weights of ribosome, then only he or she can answer this question. So what I'm saying is the eukaryotes generally have 18S rRNA, which is a part of 40S, a small ribosomal unit, while 5.8S is also a part of eukaryotic ribosomal thing, that is a part of the large subunit. The 28S rRNA is also a part of eukaryotic large subunit of ribosome, 60S large subunit. But the 5S, 30S, 5S, 23S, and the 16S. So 5S, 23S, and the 16S are from the prokaryotic ribosome. So this is the prokaryotic ribosome, and this 28S, 18S, and 5.8S are from the eukaryotic chromosome. So I can directly tell from this. This is just a data to remember. So now I have to select the correct statement. The first statement says 1, 3, 5 are from the plant ribosome. So I have 1, 3, and 5. Yes, plant ribosome. Plant is a eukaryote, so it's from the plant ribosome. This option is correct. 1, 3, 5 are from bacterial chromosome. Bacterial ribosome. Sorry, I mistakenly said ribosome to chromosome. Sorry. 135 are from the plant ribosome. 135 are from the bacterial chromosome. No, because I said these are the subunits from the eukaryotic ribosome. 2, 3, and 6 are from the bacterial ribosome. So I have 2, 3, and 6. No, 6 is from. So 2 is from. So 2, 3. 18 is 18 is from eukaryotic. So 3. This option for this option gets totally wrong. And in second option, both all the three options are wrong. In the fourth option, it says that 4 and 6 are absent in plant ribosomes. Okay, 4 and 6 are absent in plant ribosome. So you cannot say so. Okay, so this is a tricky question over here. So 4 and 6 are absent in plant ribosome. So I cannot directly say that these are absent in plant ribosome. So as it's a multiple option correct, it's not a multiple option correct question. So the most correct answer over here is 1, 3, and 5. Why 4? Why this 4 and 6 are absent in plant ribosome? This option is wrong because we cannot say that particularly this 5S and 23S are absent in eukaryotes. The major ribosome association takes place over 28S and 5.8S in eukaryotes, but these 23S and 5S are also present in uh, in in eukaryotes. So I'll give an example over this. That is a chloroplast and mitochondria, which are endosymbiont prokaryotes that have evolved inside the eukaryotes, and these were also a prokaryote. So ribosomes have this 5S and 5S and 16S ribosomal subunits. So let's move on to the next question. So option A is correct over here. So this question says that a researcher incubated algal cells with fluorescent level cytidine. So cytidine is a component of DNA. Cytidine analogs that can enter cell and gets incorporated into nucleic acid. That's what it says. The cells were lysed and different membrane-bounded organelles were isolated. Okay, so so what they did is they took some algal cells and they fluorescently labeled with this cytidine, and then what they did, they lysed these cells and then they extracted out the organelles, membrane bounded organelles. So they extracted out the membrane bound organelles, and what they see, and they homogenized it. The macromolecules thus isolated from each organelle fraction were analyzed. So they analyzed this fraction. So it is telling that which organelles will show the fluorescent signal. So it's a direct question from NCERT that mitochondria and chloroplast have their own DNA genetic material, which is a double-stranded circular DNA. Why? Because mitochondria and chloroplasts are evolutionarily prokaryotes that entered into the eukaryotes. So these mitochondria and chloroplast are membrane-bounded organelles which contain single double-stranded circular DNA. So this option gets correct over here. Moving on to the next question. So this is a question from biotechnology. So here is a marker thing. So I have a vector map of GPF that denotes green fluorescence protein, which shows green color on fluorescence. When I put light over this organism, if this organism synthesizes this green protein, then when I throw light over these cells, this cell will show fluorescence. So there is a basic chemistry behind this fluorescence, which you will study further in the course, in advanced chemistry courses. But let us analyze the further things. So I have a tetracycline resistance gene also over here, and I have an ampicillin resistance gene over here. A DNA fragment containing the gene of interest was generated using Sal1 digestion thing. So I have a Sal digestion setting over the GPS and Sal digesting enzyme or the restriction enzyme over the ampicillin resistance, and the eco or digestion at the tetracycline one. So assume that complete digestion of E. coli Sal1 was performed with the ligation of product, and transformants were played. The recombinants of the gene of interest will grow on the medium. So I have deleted the ampicillin resistant part. So ampicillin resistant will be deleted. So the organism is devoid of protection from the ampicillin. So if I grow them in on ampicillin plates, this organism will die because it has lost the resistance to ampicillin. But here in case of tetracycline one, till now also it bears the resistance part. So if I grow them on tetracycline plates, this will grow. So I have the recombinants having the gene of interest will grow on the medium that contains tetracycline, containing tetracycline and are GPF negative. So it will grow on tetracycline plates and that are GPF negative. This option will be correct for this. So let's move on to the next question. So okay, this is a question from ecology. In an ecological interaction between two species, wild plants and a crop plant, why with wild plants compete with the crop plant for the nutrition. So there is a direct negative impact on this. So there is a direct negative impact on this crop plant due to the wild plants over here. So there is a group of plant pests P that infect crop plants and damage them. Further, wild plants X1 support the population of insects T1 that feast on plant pest P. So I have the wild plant X1 that is supporting a population of insect that is feasting on plant P. So plant P is damaging. So I will say here I will say plant P is damaging the crop plant. X1 is producing X1 is supporting insects that feast on P. Next I have X1 and X2 that negatively affect crop plant. So there is a competition over here for food. Next I have further wild plant X1 supports the population of insect T1. So this is T1. Wild plant X2 support dwelling of pollinators insect T2. So there is an insect T2 which help in crop production. So because there is it is a pollinator, so it is directly helping the crop. So let us come to the statements over here. The first statement says that there is a direct negative interaction of Y with X1 and X2. That's what the competition is going over here. So this option is correct. The second option says that the interaction between T1 and T2 insects is commensalism. No, T1, T2 is not anyway related to each other because this thing is feasting on plant P and the T2 is doing pollination of crops. So they are not directly related to anyway. So this is no commensalism happening over here. So this option is wrong. I have I have the third option, which says that removing wild plants X1 and X2 from the ecosystem will improve overall the crop productivity of Y. Okay, so here I have the X1 and X2 both, but the X2 is infesting or dwelling pollinator or insects T2, that is promoting the pollinator of crop plants, that is promoting the crop production. So by removing the X2 plant, the overall productivity of crop Y will be reduced. It will not increase. So this option goes wrong. The fourth option says that the interaction between T1 and crop plant Y is parasitism. So parasitism is something that that is negatively impacted. So if I see T1, T1 is a further wild plant is supporting T1 that fest on plant pest. So the statement over here says doesn't say any relation of the wild plant X1 with the population of insect T1. Rather, it says that the T1 is festing on plant pest P, which is directly saying that these two are predation. So I cannot directly comment on the interaction between T1 and the crop plant Y. So there is no relation between the T1 insect and the cropland Y. So this is wrong. Rather, this T1 is helping cropland in increasing the productivity because it is infesting on the plant pests. That is actually damaging the cropland. Moving on to the next question. I have this question which is from evolution, which is that the four limbs of wall bat and cheetah. So this is a common question from evolution chapter. This suggests that the forelimb bone in these organisms developed from a common ancestor, and they were adapted for different functions. The most appropriate option that captures the evolution of these four limbs of the bones is. So first thing what comes to mind is so they have a similar structure. So similar structures, so and the functions get diverged when they are moving on to the evolutionary scale. So they have a common ancestor where the anatomical structure is preserved but the functions get changed. So this is an example of divergent evolution. Also, this is a case of homology because the structure. So, what is homology? The structure is same, but the functions are different. So, let us check over here. So, I have a divergent, convergent, convergent. So, convergent option gets eliminated. But in the other hand, I have something called orthologous over here. So this option B gets eliminated, option D gets eliminated. I have orthologous and paralogous over here. So this is not what is not mentioned in NCERT, but you should be aware of this particular concept of orthologous and homologous chromosome. The orthologous and paralogous genes. So what is orthologous and paralogous gene is orthologous genes are genes in different species that evolved from a common ancestral gene by speciation. But the paralogous genes are genes that are related by duplication within a genome. So these things, this is a concept which I would like to explain further in any other time. But let me tell that orthologous genes means orthologous genes means so these genes are evolved from a common ancestral. So these genes are evolved from common ancestor. So I can see that the similar anatomical structure were diverted from each other. So this particular and the anatomical structure is preserved. So this particular gene is an orthologous gene because because the functions, but because the anatomical structure is preserved over there, or the genes is preserved over the common ancestor. So this option of divergent and orthologous is correct over this question. So option A is correct over here. So let's move on to the next question. Okay, this question is from the tree of life. So it's directly saying that why viruses are not included in the tree. The reason is simple. Viruses doesn't have a perfect cellular structure. Hence, drawing the evolutionary tree with other cellular forms is not possible. This is a simple option, so option A gets correct over here. So there is other classifications of virus. So virus we have put in an we generally put virus in animal kingdom, but that's not a correct way of studying viruses. Viruses should be studied separately, so it's a different class to study. So because they do not show any metabolism, so inbuilt metabolism over here, they do not have any active genes. They only show they only show metabolism when they are in any host cells. So they do not have a minimal cell structure that is required for a thing to be said to be live. So when we say a structure to be living, when it has a metabolism, when it has a consciousness, when what is consciousness? It's the response to external stimuli. So this only comes when there is a metabolism. Viruses do not have such cell structures. So we cannot put into the branching where we have the cellular organisms present. So this is an odd and out of this particular evolutionary tree. So viruses we have not classified them under the tree of life. So this option gets correct. So moving on to the next question, question number 15. So this again a question from uh, um, evolution, which says that there is a chloroplast of certain algae taxa such as Euglenophyta are surrounded by three membranes, while Heterocontophyta and Cryptophyta have four membranes. So I can say that there is a membranal difference between these two particular class taxa of algae. The further question says that the chloroplast present in these organisms is a result of secondary endosymbiont events. So this chloroplast present in organism has entered into the organism by secondary endosymbiotic event. All of them have chlorophyll A. However, Euglenophyta uses chlorophyll B as an accessory pigment, whereas these two uses chlorophyll C as an accessory pigment. Assuming gain of function and gene addition, the most likely evolutionary scenario. So again, these questions are generally the next questions are generally asked about the best describing things. So you need to analyze the statements over here and pick out the most correct option. So let's go to the first question, first option, which says that while Heterocanada and Heterocontophyta and Cryptophyta has had a common secondary endosymbiont, Euglenophyta evolved from an independent secondary endosymbiont. This is clearly from the fact that the membrane structure over here are different, and the utilization of chlorophyll is also different over here. So this option directly gets correct. Again, I am saying the Euglenophyta is evolved from a different thing because it has a different membrane structure and it is dependent on different type of chlorophyll. So Euglenophyta uses chlorophyll B as a secondary pigment or an accessory pigment, but these two particular algae uses chlorophyll C and they have a four membrane structure. So they are not related to each other. They are coming from a different lineage, but both are affected, both have evolved from an independent secondary endosymbiont. So this option gets correct. The second option said that had all secondary endosymbiont and all had a secondary common secondary endosymbiont. No, this is not correct. Evolved from an independent secondary endosymbiont event. No, this is different because these two are at the same thing. These two are evolved from the same parental generation. And this cryptophyta were result of secondary from the Euglenophyta. No, this is wrong. So this option, this is the most correct option describing the event. Moving on to the next question. So this is a question from body fluid and circulation. This says about dengue virus, but this is a direct application question from the platelets. What does platelet do? platelet have clotting factors that stop bleeding. So in case of dengue viruses, what happens is there is a leakage of blood vessels from the leakage of blood from the blood vessels. So this is done by transfusing platelets into the patients. So what does platelets do? Platelets have the clotting factors that stops the internal bleeding. So this option gets correct over here. So I have the 17th question. In this question, this is a question from ecological interactions. Scientists have recently found that archaebacteria belonging to the phylum, this particular phylum, Leuciarchaeota, can grow only in co-culture along with methanobacterium and another aerobic proteobacterium. So this particular phylum is dependent on two things. One is methanobacterium, another is proteobacterium. Again, it says the archaeon catabolizes amino acids that are secreted out from methanobacterium producing format. The format released from the archaeon is utilized by the methanobacterium. So this is an mutualism. This was an mutualism. So the further it says that uh, the proteobacterium, by scavenging oxygen in the environment, provides an anaerobic environment for the archaeon. This interaction between archaea and bacteria could have resulted in evolution of the present-day eukaryotes. So if I talk about the proteobacterium, here I can there is one comment over the proteobacterium, that this proteobacterium produces an anaerobic environment by scavenging oxygen. So only the phylum, that is the Lochia is benefited over here, but the proteobacterium is not benefited. So this is a one-sided positive mutualism. So this we can put as a commensalism. So I have option A, mutualism and commensalism over here. So moving on to next question. Okay, this is a question from biomolecule. So what are non-reducing disaccharides? Non-reducing disaccharides are the molecules which do not have the epimers available to get reduced. So sorry, epimers, not epimers, the anomeric carbon to get reduced. So it depends on the reaction how this disaccharide is formed. So let me so what is an anomeric carbon? It is that what is the anomeric carbon? Anomeric carbon is that carbon which is directly attached to the aldehyde or the ketone group that is forming the chain, that is forming the ring, not chain, that is forming the ring. So this is the anomeric carbon side. So if I check for these if I check for these all four different disaccharide, I can see that here I have a 1,4 linkage, here I have a 1,1 linkage, here I have a 1,4 linkage, here I have a 1,4 linkage. So in A, C and D condition, I have one anomeric carbon, that is one position empty of each one, which can react to which can be a reducing sugar, which can be reduced. But in case of the second one, both the anomeric position is occupied in the bonding. So both the anomeric carbon are occupied in the bonding. So this becomes the, you can see here 1 and 1 are involved in the bonding of are involved in glycosidic bonding. So there is no place for reducing, so there is no site to reduce the sugar. So this option gets correct. Moving on to the second last question. So this question is directly taken from NCRT statement, which says that, so this is an easy, so you don't go through the question. If I directly see the options, it says that kingdom Protista, fungi, Monera and Plantae. And when I read these statements, that it has thread-like structures, branched, okay, it has cross walls, distinct nucleus, staining cell reveal the structure is complex polysaccharide. Fungi have chitin cell walls, and end up some branch structures, small spherical structures with it. So this is a common, this is a common sense to this question, because it's directly saying about the fungi, because this type of morphology is only seen in fungi. So I can directly check, go through the option and correct, and check the correct one. So there is no need to go through the whole question. Just take the important lines over here and identify in such type of question. So okay, these type of questions are elimination based questions, so you directly eliminate options and you'll get the correct options. So this is a description about the fungi, and which is a phylum from the Ascomyta, or the sac fungi. So this is the last question. Okay, so this last question is based on uh, so this question is taken from biomolecular enzyme kinetics. So you know that while when we add enzymes, the activation energy gets lowered. So I have the so this is a normal reaction, and this is the enzyme linked reaction. So the activation energy is lower. So this is the energy of the transition state over here. So this is the energy of the transition state over here. So when we add enzymes, the efficiency or the activation energy is decreased, and the substrate gets easily converted into product. So we'll check with the options. So here is the first option says that L, M, and O represent the potential energy of substrate, transition state, absence of enzyme, transition state, in presence of enzyme, and product. So I have this, and M. So it is M. So second transition state in absence of. Okay, so this is correct. So this option is correct. The second option says that the rate of catalyzed reaction is increased exponentially to tune a difference between M and N. So this is what happens. The reaction gets catalyzed in an exponential manner. So this option is also correct. The third one says that the rate of catalyzed reaction is directly proportional to the